So welcome everyone to the our 42nd Great Lakes Collaborative webinar series, our Students of Phrag webinar. My name is Ned Willig from the Great Lakes Commission, and I'll be hosting the webinar today on behalf of the Great Lakes Phragmites Collaborative. Our presenter today is Rae Robinson from Utah State University, and she will be presenting on the effects of seed mix composition, sowing density, and seedling survival on plant community reassembly in the Great Salt Lake wetlands. Rae is originally from Baltimore, Maryland. She received her BS in biology from the College of William and Mary in Williamsburg, Virginia, in 2015, before heading out west to work a series of seasonal jobs. These included trail building in Arizona, vegetation monitoring in the Oahe Mountains, I don't know if I said that right, in the Idaho, Oregon region, as well as environmental education on a small farm in Northern California, and doing stream management monitoring for the BLM on the western slope of Colorado. Rae completed an AmeriCorps Vista year at Rivers Edge West, which is a nonprofit that focuses on riverside restoration. Before that, and then in 2018, she headed to Logan, Utah to pursue her master's degree in ecology at Utah State University. As a student in the Wetland Ecology and Restoration Lab, Rae is focused on revegetation of native plant communities in the Great Salt Lake wetlands. And today we will hear about her master's research. So Rae, thank you so much for sharing your work today. I will now transfer presentation control over to you, and feel free to take it away.
Okay, thanks, Ned. I'm just gonna share my screen.
Okay, and it looks good, so you're free to go.
All right, hopefully everyone can hear me and see this presentation. But yeah, thanks for the introduction, and I'm excited to talk to everyone about some of the research I've conducted these past couple years. So I'm going to talk a little bit about Great Salt Lake wetlands and the need for seed-based restoration. Then I'll move into my research that focuses on seed mix composition and sowing density. And then also fine-scale demographic data collection looking at germination, emergence, and seedling survival. And then I'll end with summary and conclusions and also some future directions. So the Great Salt Lake is the largest terminal lake in North America. It's located in northern Utah. And like a lot of wetlands everywhere, it's at risk of further degradation, and it's associated with a whole laundry list of environmental challenges. It makes up about 75% of Utah's wetlands, and it's of continental significance because of its location along the Pacific and Central America's flyway. So over 250 migratory bird species rely on these wetlands as a place to rest, cover, and also for food. On the right, I'm showing this table of some of the priority bird species on the Great Salt Lake. And I like this table because it shows the different wetland habitats that these birds rely on. And so the different colored squares there relate to submergent, emergent, meadow, and playa wetlands. And so these different wetland types are associated with plant communities and also flooding regimes. And unfortunately, maintaining this mosaic of robust native plant communities is difficult because of Phragmites, which threatens wetlands across North America and also on the Great Salt Lake. I'm sure many of you know it replaces native vegetation and open water, and it spreads prolifically through clonal growth, but also through robust production of seeds. And it negatively impacts avian habitat. Studies have shown that stands of Phragmites are associated with lower abundance of waterfowl and shorebirds. There's also been reported shifts towards smaller bodied generalist species, and also lower avian diversity scores in monotypic Phragmites stands. And that last citation there is compared to other native monotypic stands in Great Salt Lake wetlands. There's been quite a bit of research to determine the best ways to control Phragmites. And so treating Phragmites with herbicide, grazing, mowing, cutting, crushing. So lots of these different treatments. over multiple years has been shown to be effective in terms of killing Phragmites. But unfortunately, native plant recovery is highly variable and often slow. So if you look at this picture on the left here, this is after Phragmites has been removed. There's a dense litter mat, which is problematic for some of these native species that are regenerating from seed that need high light conditions. And then you can also see the vegetation in the background, this photo here. That's just Phragmites coming back. So there's competition with not only Phragmites, but other non-native species that have been reported in and around Great Salt Lake wetlands. And there's also limited native plant propagules, which is a result of these wetlands that exist in this degraded condition, disconnected from robust native plant communities. So for all these reasons, we need active revegetation to restore some of these native plant communities on the Great Salt Lake. So seeding is logistically feasible in that you can cover a large area. It's cost effective, and that's in comparison to coir mats or plugs, which might be useful for like smaller targeted restoration areas. Seeding is also a way to influence ecosystem functioning because you can select species that help achieve certain restoration goals. It's also a way to increase genetic diversity, which we know is important because it provides a strategy for plant species to adapt to change. And it's a good strategy for heterogeneous, dynamic wetland environments. So the idea is that you can form a seed mix with different species, with different traits, different hydrological tolerances, and sow that mix across the wetland. And that's a bed hedging strategy that you'll at least get some native plant establishment. So even though I just talked about how seed-based restoration is definitely promising, in wetlands in particular, it's challenging. So these are already degraded and disturbed ecosystems with a heightened threat of invasive species, which is partially attributed to the location of wetlands within the watershed. They're hydrologically dynamic, so that's one way that it affects plant establishment, but also oftentimes if you're sowing seeds in a wetland, even if you use some type of tackifier or botanical glue to keep the seeds in place, that might degrade and your seeds might just move away from your restoration area. And after these sowing efforts, we expect that population mortality is high. And there's studies from other upland or non-wetland systems that have shown that there are these really big germination and emergence bottlenecks. So to talk a little bit more about these plant stages here, if you look at this diagram, so germination we can think about as when the early embryonic root pokes out of the seed into the soil. Emergence is when the seedling pops above the soil surface. Establishment is when the seedling is now its own autotroph, so it's photosynthesizing and no longer relying on the seed reserves. And then survival, which can be defined as survival to a certain point in time, like the end of the growing season or after a couple weeks. And so one group of researchers found that only 7 to 17% of germinated seeds make it to emerge seedlings. So that's a huge population bottleneck there. And for wetlands, we need to quantify these life stage transitions. And you can imagine how in wetlands, the drivers of these systems might be different than compared to an upland system. So in wetlands, conditions might be too dry, but they also might be too deeply flooded. And so you could imagine how that would affect the earliest stages of plants' life. My research questions, the first one, what are the effects of seed mix composition and sowing density on plant community reassembly? And I'll talk through some of the experiments I've conducted. And my second question, how do species and environmental conditions affect germination, emergence, and seedling survival? And I'll talk through those experiments as well. First, what are some of the native plant species that we're interested in restoring? They're shown on the screen here. They exist across a broad hydrological gradient. We have native bulrushes, which are important because of their habitat value. We have a spike rush, an Arctic rush, 2 grasses, and then some forb species as well. Background information for this first part of my research question, what is the ideal seed mix composition? So compared to upland systems, research is limited. And the species I'm showing on the screen here are habitat-forming perennial graminoids, and graminoids is like grass-like morphology. And so these species here, either a few or several of them, are typically used if seeding is attempted in Great Salt Lake wetlands, because these species are the preferred habitat for waterfowl, and therefore they're the preferred habitat for some of the wetland managers that are attempting these restoration efforts. And in this research, we're interested in investigating some of these forbs that we suspected when we started this research to be fast growing. And the idea is that if you can add these forb species to the mix, we're asking, can you get better invasion resistance by emerging sooner and limiting light for these Phragmites seedlings, which we know are also quick to emerge. Some research has shown that, yeah, actually annuals can outcompete Phragmites better than other plant functional types. But other research has shown that if you add these fast-growing forbs to the mix, they can actually suppress the species on the left. And so for some wetland managers, that might be problematic if they want some of these habitat-forming perennial graminoids, specifically for their functional value or some of their restoration goals. What species quickly establish and preempt these resources, and ideally, ultimately, what is the best seed mix to increase native cover and decrease non-native cover? And then the second part of my question, what's the ideal sowing density? Again, research is limited compared to upland systems, and the recommendations that do exist are variable. So if you look at this diagram on the bottom, I don't expect you to read everything on there, but I just want to impress upon you that there's a large range of recommendations, but also things that have been attempted in different experiments, ranging all the way from passive revegetation, so that would be like a 0x sowing density, a somewhat standard base rate recommended for Utah wetlands, which is a 1x density, and then 3 and 5 times that base rate, which other research in my lab conducted in mesocosms has shown that those higher densities are associated with reduction in Phragmites biomass. But it's going to be important that we test these densities in the field. Another aspect of this question is, are density-dependent effects evident at higher densities? So it's important for us to identify if there is a threshold at which sowing more seeds actually won't provide more benefit. And so this is important because managers are operating with limited capacity. So we want to make sure if that we're recommending to sow more seeds, that comes with added benefit. So here are some of the field sites that we conducted these experiments over 2 years. So they're basically sovereign lands on the Great Salt Lake lakebed adjacent to these 2 waterfowl management areas. And I've listed on the right the Phragmites management treatment history in case anyone is interested, but basically multiple years of treatment, which we are recommending before revegetation is attempted. In preparation for the experiments, we secured seeds from our partners and also collected them from the wetlands. And many of these species exhibit dormancy, which is a characteristic that prevents germination even under ideal conditions. And so to break that dormancy, we have to either apply a cold stratification or some type of scarification treatment. So I just wanted to mention that was all done in preparation for these experiments. The first thing we looked at was 2 different mixes sown in 3 different densities in the greenhouse. So our 2 different mixes, one was a 4-species mix, which is representative of maybe a more typical sowing approach. And then we also tested this experimental 9-species mix. So it's those perennial graminoids plus some of those forbs. And some results for everyone. So to orient everyone to these plots, on the x-axis we have the day of the experiment. On the y-axis we just have the cover proportion. And you can see that even at day 15, so only 2 weeks into the experiment, we're seeing really large differences between the 9-species and the 4-species mix. So the 9-species mix is very robust, and you can see a lot of those forbs there in the picture with the broad leaves. We also saw not only mix effects, but density effects. So in general, the 3 and 5x densities had higher cover than the 1x. By the end of the experiment, most of these mixes and density combinations reached high cover, with the exception of this 4 species mix in the lower density that kind of just lagged behind. And I will mention here for the 9 species mix, you can see from this photo taken at the end that there's not a lot of perennial graminoids in here. It's mostly just those forbs. So if they're, as I mentioned before, this might be important for some managers that actually care about establishing certain perennial graminoids. So what did we learn from this experiment? We saw there was mixed differences. We saw that, yes, some of these forb species that we suspected to have robust growth forms and to quickly emerge, they did that, and they suppress some of the perennial graminoids in the process. And then we also found density effects where the 3x and 5x densities were higher than the 1x, but the 3x and 5x weren't significantly different from each other. This led us to our next question. Well, that's great that we examine this in the greenhouse, but how do these mixes and densities perform in the field? So last field season, we set out to look at 4 different mixes of native species across 2 different densities in these 1/4 meter squared plots. And now these mixes are different than the greenhouse. So the mix 1 is 0% of those fast growing native species. So those are on the right here in this table. So those are 3 of the forbs, and 100% of the perennial graminoids. Mix 2 is a 1090. Mix 3 is a 2575. And then mix 4 is kind of the other end of the spectrum. So just 100% fast growing native species and no perennial graminoids. So we sowed these plots in June and took percent cover across time. And here are some photos that we took at the end of the growing season. So I will talk everyone through this. On the first row here, I'm showing in the 3x density these mixes that had more perennial graminoids. And you can see that from the photo, there's a lot of grasses in there. I see some hard stem bulrush. On the bottom row here, I'm showing the 100% fast growing natives mix, which actually a lot of this biomass had senesced by the end. So it kind of looks, it's red or brown and kind of crispy there. And then the control plot, which was not seeded, you can see just a lot of cattail in these plots. So we did a preliminary ANOVA to just see, looking across the different densities, is there a difference between native cover? And we found that yes, there were significant differences between the 1x and 3x had significantly higher native cover than the 0x. So that was expected, and we wanted to continue to analyze this data to further investigate our mix and density effects and look at this across time. So that's what I'm showing in this plot here. And there's a lot to look at. So on the x-axis, it's a time series. It shows July through September. And then on the y-axis, I'm showing native cover, which is a sum cover category of all those natives of interest that we sowed. We have our 2 field sites as the 2 columns, Farmington Bay and Howard Slough. On the first row, we have our 1x density, and our second row, we have the 3x density. And you can see that cover begins to increase across time, and it kind of peaks around early August, and then some of these mixes actually start to senesce. So by the end of the growing season, we actually saw that the 0, 10, and 25 fast-growing natives mix, which are the mixes with more perennial graminoids, actually had higher cover than this 100% fast-growing natives mix, which you can see, for example, at Farmington Bay. Though the mix peaks in August, it does quickly senesce. So we did see some mix differences. We also saw some density differences. So the native cover of the 3x treatment was significantly higher than the 1x, and that was associated with a 20 to 50% increase from the 1x density, which comes out to about 14 to 18% difference in cover. This is opposite of what we predicted and what we would have thought after conducting our greenhouse experiment. So why is it that we saw that the perennial graminoid mixes did better than these fast-growing forb mixes? And I think it has to do with the variable species performances. So Distichus spicata or saltgrass dominated these perennial mixes. And of the fast-growing forb species, we only really saw this Biden cernua consistently, which helps to explain why we saw what we saw. All right, now I'm going to ask everyone to kind of reverse their brains here. So this is the same experiment, same plots, except now I'm showing on the y-axis non-native cover, which is a summed cover class or cover category of rabbit's foot grass, Phragmites, and also cattail, which cattail is probably native in the system, but it's considered to be undesirable and invasive. So for our purposes here, we're just considering it as a non-native. And you can see that across time, non-native cover also increases in these plots. Though we do see in the 3x plots at both sites, we get a reduction of non-native cover at that higher native sowing density. So that's a anywhere from 30 to 55% reduction in cover. And even though the higher sowing density reduced this non-native cover, cattail species were the most prominent of the non-natives that we were recording, and it's still established among our seeded natives. So I think what happens is cattail is a very strong competitor. It emerges early and then is able to establish this robust cover that kind of exists like above some of our seeded native cover that exists closer to the ground. To wrap up this experiment for everyone, what did we learn? So we saw the biggest differences in native cover between the 0x and 3x densities. We also saw that there was added benefits of the 3x density when compared to that base rate, the 1x, and that was associated with significantly higher native cover and lower non-native cover. We found in terms of mix that the mixes with more perennial graminoids had higher native cover, and that was mediated by saltgrass. Actually, so saltgrass and Puccinellia nataliana were in a joint cover category, and we think it's mostly saltgrass, but we're going to monitor year 2 to follow up on that and see if we can tell the two apart. And the performance of this forb species was variable. So as I mentioned before, nodding beggartick did really well, but these two other forb species did not do very well at all. Like we rarely saw, for example, the fringe willow herb. And I don't really know why that is, why they did so well in the greenhouse and then didn't do well in the field, but it probably has to do with some aspect of the field environment. Both golden dock and fringe willow herb are smaller seeded species, so maybe they were disproportionately affected by some aspect of the field. And I think it's important to also consider phenology or seasonal timing when assembling these native seed mixes, because even though Biden cernua could establish high cover in August, it quickly senesced at some sites. And so when assembling a seed mix, considering phenology would be one way to ensure that you get high native cover throughout the growing season. All right, I'm going to talk about just this last experiment for this first research question here. So here we weren't looking at mix, we were just looking at density in more realistic field conditions. So we sowed this one mix of these species in relatively equal proportions into these 25 meter squared plots. So these are much larger plots than the experiment I was just talking about. We broadcast these seeds. That's me doing that in the right hand corner here. And then we sprayed water and tackifier, which is that botanical glue, on top. And then we monitored percent cover throughout the growing season. So to orient everyone to this plot, on the x-axis, we have our time. On the y-axis, we have our cover. So the first row there is showing natives, and the second row is showing non-natives. The natives is just the summed cover of those natives of interest that we sowed. And the non-natives is the same 3 species from before, though we saw mostly cattail. Definitely, yeah, a lot of cattail. So what did we see? We saw at Farmington Bay for native cover does increase throughout the growing season, and we are seeing the 5x that has higher native cover than the 1x and 0x, though the 5x and 1x are not significantly different from each other. So we are seeing the biggest differences between the 0 and 5x. And then in terms of non-native cover, we're seeing, if you look at Howard Slough, for example, the 5x density is associated with the greatest suppression versus the 0x density there's a lot more cattail in those plots. So even though we're seeing some density effects here, they're more subtle. And it was surprising to see that there were subtle density effects, considering the last experiment I was talking about was looking at 1x and 3x, and this experiment is looking at 1x and 5x. So why is that? I think we lost some seeds after we sowed, so that definitely could have affected our results. And there was a larger plot size. There was some remnant litter left over. Also, we used a different seed mix compared to the other experiment with different seed sources. So all of these things could have affected our results. And even though we don't see significant differences between the 1x and 5x densities, I think it's important to note that we're still seeing the same trends, where the 5x is associated with higher cover and greater suppression. So to wrap this first question up, in the greenhouse, we saw that fast-growing forbs suppress perennial graminoids, but in the field, we did not see that. In fact, mixes with mostly perennial graminoids perform better. And this is attributed to the performance of some of these species in the field. And so I've kind of ranked them from high performing to low performing in the field in case that's of interest to anyone tuning in today. We also saw added benefits of the 3x and 5x densities compared to the 1x density. And when we started this experiment, we weren't sure if we were going to see these strong density-dependent effects where higher densities don't have added benefits. But in fact, we did see that higher densities do have associated benefits. Okay. And just before I end here, I'm going to just briefly mention one experiment that I conducted in the field looking at the effects of species and environmental conditions on these early life stages. At this point, you might be wondering, so why are you tracking these seeds and seedlings? Like, isn't that overkill? But I think it's important that this research is important because determining these population bottlenecks will allow us to then develop strategies to overcome them. So to look at germination and to get these germination proportions, we put seeds of these species in these little baggies and pin them to the soil surface. A lot of these wetland species need high light, so this is an appropriate technique. And then for emergence, we seeded these containers with a set number of seeds and then tracked the presence of a seedling with these labeled toothpicks. And then weekly, we checked to see presence absence, is the seedling still there? And we did that for 4 weeks in the field. So what do we find? Here's a plot of our germination and emergence data for a lot of these native species and then also Phragmites. So first of all, each species has its own little square here. The two sites are shown in the different colors. Germination is the first column here, shown with these little boxes, which is basically the number of seeds germinated divided by the number of seeds that we put in the germination bags. And then seedlings, week 1, 2, 3, and 4, it's just number of seedlings there divided by number of seeds that we sowed. The viability, which is like the proportion of that seed source that's alive, according to our laboratory tests, is shown with this dashed line here, so it's different for each species. What's striking to me is that, some of these species don't do very well at all, so some of these bulrushes down here have lower germination and emergence. But some of our species, like Bidens cernua, Rumex myrtimus, and Disticus vicata, actually do relatively well. For Bidens cernua and Rumex myrtimus, the ND stands for no data for germination, because we think these species quickly germinated and then they decomposed before we could grab the bags from the field. But you can see how for a lot of these seedlings, they increase throughout time and then decrease. Actually, our field notes for Bidens cernua points to desiccation at Farmington Bay, one of our sites. They emerge and then basically survive throughout time. And so here are high-performing species. As for the rest of these species, what do we do? So I think some of these that have low germination suggest they have deep dormancy. So more intense dormancy breaking treatments can improve germination. And even though we set out to investigate how environmental conditions affect these species and these life stage transitions, we actually weren't able to incorporate conditions into our statistical models, but we can still speculate as to how these conditions might have influenced our results. And I think dry conditions likely limited germination, emergence, and survival thereafter. So what do we find from all this seedling tracking in the field? So sowing these high-performing species that we identified on the right here may optimize our native cover. And then as for the natives that have inconsistent or low performance, that indicates the need for higher sowing density, particularly if, as I mentioned before, there might be a species that is definitely desired for its ability to help achieve these restoration goals. And so in that case, a higher sowing density could help mediate or assist in that species recovery. And then also other interventions. So more intense dormancy breaking treatment for low germination, like the bulrushes likely require more intense treatments. And there's also emerging seed coating technologies that are maybe could be helpful. Lastly, avoiding conditions that are too dry or too deeply flooded might also help. So to wrap up all of this research for everyone, the scale of restoration and the need for restoration is quite large. And our research suggests that when seeding, rates of 3 and 5x are justified compared to that base rate or relying on passive recovery alone. Seeding a multi-species seed mix is one way to ensure some native species establishment, in particular, including the species that we know can help optimize native cover, like the saltgrass or the nodding beggar tick. Maintaining moist to shallowly flooded conditions is ideal, as these more deeply flooded or too dry definitely probably limits those early life stage transitions. And multiple years of Phragmites treatment is also helpful in terms of allowing for the natives to just have a chance to establish. And so in my first research, in a lot of my research plots associated with my first research question, we actually saw a lot more cattail than we saw Phragmites. And I think a lot of that is attributed to just the multiple years of treatment at our restoration sites. In the future, as I mentioned, we're going to do long-term monitoring, definitely this year and maybe future years, to see if these natives are still present in the plots. I think we need to focus on improving water control and keeping seeds in place because that likely affected the results of at least one of my experiments. Pursuing techniques to fully unlock the full potential of species from seed, and that's that improving dormancy breaking techniques and also pursuing emerging seed coating technologies that might help improve the emergence of germinated seeds, for example. And also continuing to explore other species, other mixes, and other functional traits that might help catalyze resource preemption and prevent the reinvasion of Phragmites. And so there's other people in my lab that are looking at different species and mixes. With that, I'd like to thank everyone that supported this research and take any questions you all may have. Thanks.
Awesome. Thank you so much, Rae. That was really great. We will, if anyone has any questions, feel free to put them in the chat box there, and we'll take a look at them. I guess the first question that I had, Rae, was, is there a difference? Could you talk about like just the difference between the two sites that you looked at? It looked like there was some slightly different results that you found, I think, between the Howard Slough and the other place. I can't remember what the name is, but what are the differences there?
Yeah, so this past field season, there was definitely differences in the standing water depth throughout the growing season. Mm-hmm. And so, yeah, I didn't talk about that in this presentation, but basically at Farmington Bay, there was more drawn down saturated conditions. Mm-hmm. And then at Howard Slough, there was a little bit more standing water. And I think for a lot of those multi-species plots that I was talking about in the beginning of the presentation, there was a lot of cattail. And I think that standing water at Howard Slough definitely perpetuated more non-native / cattail growth in those plots. Gotcha. So, right, that, yeah, and I think we do know that hydrology is a major driver of plant community assembly in wetlands. And so it makes sense that that definitely affected my results. Mm-hmm.
Nice. That makes sense.
And that's, I also measured another condition that we were interested in when we started this research was salinity. So I also measured surface water salinity when present. And that was typically no greater than 5 parts per thousand if you think of seawater as like 30 or 35 parts per thousand. So it's pretty fresh. And so we don't think that was a major driver in some of our results. And that most of the wetland plants we study probably could have tolerated that salinity.
Will the intent as you move through, it sounds like this is going to be a long-term project and this was the first start of it. Will the intent to be to track the different conditions to see just changes in salinity and water saturation and whatnot, how those affect the plots as they go along?
Yeah, I mean, ideally, yeah, that makes sense what you're suggesting. We're actually it's still in the works as to what the trajectory of these plots will be. I know I'll be monitoring them again this field season. We don't have any long-term environmental condition plans right now, but I guess that could be subject to change. I think part of it's interesting, like we're still trying to figure out the best way to monitor these conditions in the field because we come out typically once a week. And in that time, sometimes when we get there, it's clear that like the water level rose and then fell. And so, yeah, the scale that we're collecting this data also, I feel like, is important. And we haven't really figured out the best way to do that yet.
Yeah, so we have some other questions coming in. What effect do you think soils used in the lab experiment versus the field experiment play? And did your fast-growing annuals do well in the lab because you used potting soil?
Yeah, I mean, that could be the case. Right, so certainly the potting soil that we use in the greenhouse and then in those life stage tins. So like potting soil was in those tins and then we put those tins in the field. Yeah, definitely different. And we've thought about that a lot. I mean, in terms of, it was important for us to collect this fine scale demographic data. And so with other species coming back from the seed bank and competition affecting those life stage transitions, we were more so trying to just capture just the abiotic conditions with the species being mostly competition free for that life stage tracking. But right, in a more realistic situation, you're going to have competition, you're going to have soil microbes and pathogens that might attack your germinated seeds or your freshly emerged seedlings. You might have, right, it won't all be nice and smooth and saturated. There could be litter. Yeah, so that could definitely affect results for sure.
Definitely. One other question was, I think you had mentioned this, but if you could just go over again how the Phragmites were removed and how soon after Phragmites, the management of Phragmites were the seeds applied.
Yeah, so I conducted experiments across 2 years. So it was and then across the different sites, there's different treatment histories, but it's anywhere from 2 to 5 years of treatment. And that was typically a herbicide spray in the fall, I think it was August, and then some type of spring, either grazing or mowing. And then some of these sites too also had some type of cutting technique also into the mix there. And so that slide listed like all the specific treatments. So I'm happy to share this presentation in a PDF form, either to you, Ned, or if people want to email me, that's fine too. I can send it to them if they're interested in like the specifics.
Yeah, that would be great. We would definitely appreciate. I can share that out if you want to send that to me. We can in the post meeting materials. That would be great.
Okay. Yeah.
Another question about the seeds. Do you know where the seeds were sourced from? Could you talk a little bit about that?
Yeah. So a lot of this, so some of those all of those fast growing forbs we collected from Great Salt Lake wetlands, just on the eastern side of the Great Salt Lake there, because most of those species are not commercially available. At least they weren't when I started these experiments a year or two ago. And then a lot of our bulrush and saltgrass species we got from our government partners that have a big seed warehouse here in Utah. And those were collected throughout wetlands in the Intermountain West.
Gotcha.
I believe.
And so then thinking about the seeds, was resiliency to climate change considered in any of the sourcing of these mixes, or any thoughts related to the impacts of climate change and how those were considered?
Yeah, this is something that we talk about in the lab every so often. So a lot of these species that we sowed are, and a lot of these forb species in particular are meadow plant, are associated with a meadow community. So like a drier, less deeply inundated, less frequently inundated area. And so we think, yeah, in a hotter, drier climate or less consistent access to water in these wetlands, that might be advantageous to include those species that can exist in a drier context.
Yeah. Oh, that's great. Yeah, that's really interesting. Do you think and so another question we have here is, do you think after the annuals die back, it will make way for perennials that are in your mixes? And so start with that question.
Yeah, for sure. And that's a reason why we wanted to monitor this coming year. We were wondering if we're going to see more bulrushes this year, especially given the deep dormancy that I was talking about that some of these species exhibit. Maybe having like 2 overwintering periods would help them. And then by year 2, maybe we'll see more of them.
Okay, great. And then are you, I guess related to that, are you optimistic about the next season for the mixes that showed poor end of season results from the annuals dying back?
Yeah, we'll see. A lot of those annuals, they did set seed. I don't know if like those seeds like ended up in the plot. They might have gone somewhere else. But I'll be curious to see. You know, I was talking to one of the managers who was out there the other day, and I said, how the plots look, and they said, oh, you know, there's a lot of cattail, like we can't even see your plots. I was like, no, this is not what I want to hear. But yeah, we'll take a closer look this field season.
Does it seem like there are less Phragmites than cattail? That seems like that was one of the results you were showing. Is that significant or of note to you guys?
Yeah, definitely. I was actually kind of surprised because so the experiments I was talking about in this presentation were conducted last year, but I also conducted experiments the year before that I didn't talk about. And the year before, there was Phragmites in and around a lot of these plots. In fact, for the experiments I conducted that first year, I ended up weeding the plots because they were just like taking over the natives. And I was like, well, it'd be more interesting to weed and say we weeded and then collect native data, native plant cover data. But this year, because there was less Phragmites, we didn't have to weed. And yeah, we actually were seeing more cattail. And I think that's just a product of another year of Phragmites control that really just gave the natives a chance.
Gotcha. Interesting. Are there any further questions? We have one. Okay, so related to what I had just asked, do you know if cattails were present in high amounts before removing the Phragmites?
Sorry, was it present?
In high amounts, like were there a lot of cattails with the Phragmites before the kind of treatment for Phragmites?
Right. Yeah. So I'm not sure about that one. I would assume so. I know that it depends on the managers how they treat cattail. So some of them I know will keep cattail stands because they think like, oh, well, it's not Phragmites, so this is kind of a strategic cattail stand. But if it's mixed with Phragmites, some managers treat that just as they would Phrag, especially during like an aerial spray. Then the cattail is also being treated. So yeah, I don't know like the specifics of the cattail stand before my experiments, unfortunately. But I mean, I would guess that there was probably quite a bit there.
Gotcha. Great. Any other questions that we have? Give one last chance for folks. Any final thoughts from you, Rae, on any of what you'd like to share?
I guess I would just say I know it's a lot of information at once and it's a lot of plots and results. And so if anyone has any specific questions, they can just feel free to email me.
Okay. And we will share your email after the presentation if that's okay with you as well.
Yeah.
Great. So again, thank you so much, Rae, for doing this. This was really great and really interesting, and great for those of us in the Great Lakes to see something outside of our region and learn more. So for those that are hoping to share this with others, a webinar recording will be available in the coming days, and we will send that out to everyone that attended today. We also, if this has been an interesting talk to you, we will have an upcoming webinar from I believe a lab mate of yours, Rae,
Yeah.
Emily Tarsa, and she will be talking about seed-based wetland restoration following Phragmites australis removal, and harnessing the seed traits and systems modeling to promote native plant establishment. So we're really excited for that, and hopefully can continue this conversation more further. And that will be May 20th at 1:30 p.m. And that will conclude our presentation. Again, we'll send meeting materials out after in the coming days. And thank you again to Rae. And we want to just acknowledge the USGS for their funding under the GLRI for this presentation. And thank you everyone for watching. All right, thank you.
Thanks.
Bye.